Okay, everyone, let's get settled down. Who's ready for some bioart and design? That was kind of insufficient enthusiasm, in my opinion. Who's ready for some bioart and design? Right, that was better. Okay, so we're gonna have a very special person host this particular session. So I'd like to introduce to the stage Annie Liu. So everybody, a round of applause for Annie. So Annie is an amazing, well, first, Annie's just a wonderful human being. Let's start there. And then on top of that, Annie's also an extremely talented artist and designer. She's working on a number of really, really amazing projects. And if you kind of just look at her, find her stuff on social media, her Instagram page is also amazing. Which you should, what is your Instagram? It's at? Wondering Animal. At Wondering Animal, okay? You can find it off of mine if you want. But Annie is doing a bunch of really, really amazing projects, and I think is kind of one of the real rising stars in bioart and design and doing some really, really wonderful things. She just finished her master's at the Media Lab here and is living in New York now, and is just kind of kicking butt, doing awesome stuff. So really, really honored to have Annie here to host this session. So one more round of applause for Annie, please.
Is this on? Can you hear me? Okay, great. So it's my pleasure to introduce the EMW Street Bio group. They're talking about Biota Beats, which I had the pleasure of working on. So I know that EMW Street Bio has already been introduced, but it's literally the place where I learned everything I know about bio. Has a diverse of creative thinkers, everyone from synthetic biologists to breakdancers. So presenting this project will be Alex, who is an amazing rising junior from Wellesley College. She's a member of EMW Street Bio. Okay, sorry, I have total stage fright and totally messed up. Okay. Now also introducing David, who needs no introduction whatsoever.
Make some noise for Alex!
I'm so sorry. I'm gonna give you this mic now.
Oh, okay. Hello. All right.
Okay, well, so this is Alex. Just really quickly, I'm just going to say, as a preamble, because we got confused with our instructions, but this project, Biota Beats, came from our community lab, and it's connected basically to this question that kind of came uniquely from our lab, because our lab, our space, our community center started out as an art, technology, and social justice space, and so we have all these DJs and producers, and we participated in iGEM 2 years ago, and for our iGEM project, we were asking ourselves, like, hey, you know, what cool project could we do that's connected to our community lab? And we were surrounded by all these turntables, and somebody said, I think Mary and Verge, I think were in a meeting, we're like, hey, why don't we like do something cool with bio and like that turntable? And then that kind of connected to this larger question of, you know, hip-hop is this major cultural language in the US and around the world. And so we were thinking to ourselves, hey, you know, how do we connect hip-hop with biotech? And that's kind of how Biota Beats was born. And so I'm going to pass this to Alex to tell you more about it.
All right, cool. Okay, so there's this first question. How can hip-hop save biotechnology? Okay, so... Here's a picture, and I guess I may be preaching to the choir here, but I guess probably everyone here knows what the microbiome is, but if not, bear with me for a second. The microbiome is basically the collection of all the organisms that live on the human body, right? And I'm sure you guys have all reading headlines where the microbiome just has been, you know, the source of so much interesting research, from like fecal matter transplant to just like how it's important in all these different complex phenotypes in health. And you know, consequently, there's a lot of great research happening in like academia and institutions. But as David already kind of told the story, how can EMW, right, this place where we have wonderful monthly open mics, these like underground music DJ parties happen every month, how can this place, you know, have a different take on this question? So again, with the old record player in the room where we were trying to do bio, we came up with the question of, what does your microbiota sound like? So, yeah, we were looking at the body, and really, one of the great things really interesting things is that one of the densest place the place on Earth where there's the densest population of microbiota is in the human gut, and that's like trillions of bacteria on the scale of like 10 to the 10th, 10 to the 12th, population-wise. And I can click. Okay, so in our system, basically, you'll see on the next slide, we are working with the metaphor of DJ interaction and being able to not only listen to your music, but interact with it, right? Because this is a project not only about, well, as a BioArt project, it's about interacting with the community, right? With getting more people engaged and really understanding what is microbiome, what is the research that's going on, right? Understanding what is in your microbiome. Having a different perspective other than just like experiments and like what that could sound like. So this is the actual system we have. And so you can see there's the record player, and it was retrofitted to basically be an incubator. So there's a heating element, which on top of that is a plate, which we'll see in the next photo, is essentially based, again, to look like a record player. But it's really a large Petri dish that was custom cut acrylic, and it's like either 10 inches or 12 inches if you want to be like an LP or an EP. And we tried with 2 different designs, but the essence of it is that you have agar in it, and you basically sample different parts of the human body and put these onto these different sectors that could be either circular or pie, and you grow it. So you just go back for a second. You would grow it, and you'd incubate it over the period of time and take pictures of it and take a time lapse. And so that's happening. And then we also have the algorithm, which is what takes this image data and then turns it into music, parameterizes it, and yeah. So here is Annie. So I guess I get introduced Annie now as the wonderful member who first contributed her microbiota to our project. This is her demonstrating how we collect the microbiota. It's a very simple process. It's really just a cotton swab dipped into DI water, and you just like aggressively attack whatever body part you want. Just like dig deep between your toes, as Annie is demonstrating there. And then you want to make it like a very thorough streak over the plate. So you want to minimize, I guess, the impact of your streak pattern on the growth, because basically the features we use, I'll show you later, the data streams we collect, that matters. So yeah, you dip in DI water, you smile for like nice photos, and yeah. So this is now kind of how the algorithm works. So I guess full disclosure, we're working on different ways of doing this now, but this is what you're going to hear later on in this presentation. And what we did here is use like Python, use like OpenCV2, that library, to do some basic image processing. So the first step is to inoculate the organisms from the body and capture the images, which you just saw, right? That's Annie, yeah. And you can see here that those are some sample body parts. So you can do like the mouth, the feet, the genitalia, belly button, and the armpit. We chose these because they kind of just like form like a nice straight line down your body, just to sample from a lot of different places. Annie's also done some previous research where she kind of checked like where in your body, where's like an interesting place to sample from that you can get a lot of bacteria from. So that's kind of helped us dictate some of our choices here. But you can see, so the next step would be to do image processing to detect the organisms and to sectorize by body part. So that's basically when your computer is looking at this collection, really just pixels, and trying to decide what's the colony, like what are these things, how do I know what's what? So first you figure out that's what the colonies are, and then it doesn't really even know what the sectors are, so we need to sectorize the image and tell it that, oh, okay, just kidding, that happened. The yellow lines highlighting are showing it that like it's figuring out like those are the 5 sections of the plate. So the next step is to generate the data streams. And these are the parameters that ultimately get turned into the music. You guys have probably heard of other ways of generating, computer-generated music like genetic algorithms. And ultimately, it's all the same, where you just take in some variables and you make into new variables for music, right? The different ways music can sound with like timbre or volume, or octave, all these things, right? And those things you can change depending on what you put in. So in our first iteration of this, the main variables we worked with were angle or radial distance, and also diameter, which is, like there. I think you guys know what diameter is. And then density, which was basically the same for every section of the plate. All we did was take the area of all the colonies and then the total area of the sector, and that's the general measure we use for density here. So those turned out to be different parameters. For instance, I think we did use diameters like octave, and then I think one of our first iterations of this, at least, was that we used the 5 sectors of the plate as different notes of the pentatonic scale to kind of get it sound nice. And then the size would then dictate the octave of that note. Yeah, so that's one way you can use those data. But again, that's up to you how you want to do that. So here, you get to hear some of this music. So the output that we first generated was a MIDI file. And you're hearing right now this kind of like bass line, thanks to Chucky, who was showed later. But anyway, you can kind of hear already like some of the distinct beats in this, like the pulses, right? And those should collaborate. In essence, our idea is that every colony is a note. So it's on like loop right now, but basically that's what you're hearing. Next we have the genitalia, which is this kind of bass line again. This is, yeah, I guess I'm getting some laughs. That's the genitalia. All right. Next, the belly button, which I think here you can more distinctly hear that there's only one tone for that one colony, right? That's just pretty constant. And then you have the armpit. Yeah? Sounds pretty good? Sounds pretty good, yeah? Okay. And then last but not least, you have the mouth, which has some more stuff growing on it, and it's kind of the melody. Just like these little dots of things that are probably causing bad odors in your mouth. So, you can hear them all together, thanks to Chucky, in the symphony. And yeah, so the parts of the plate will glow as they come up. And the parts of the body as well. Yeah, so pretty cool. Yep, I guess okay. This is just everyone we need to thank for doing this this project that's been going on since iGEM. Chucky, you can see there, did the processing the music to make it sound really nice because the MIDI output is like okay. Me and Gautam were doing a lot of the software, and Sarah did a lot of the visual processing that you saw. And then everyone else, we're all doing the iGEM team, which has been on this since the beginning and made this possible. So I guess back to Congo. Yeah.
Can we get a round of applause for Alex? Yeah. So Alex did really amazing work on all the coding and every part of this. And just to kind of put the coda on what's happening now really quickly. So we were thinking to ourselves, right? So people have like vinyl record collections. What if you had like a Biota record collection? What if we could get microorganisms from like really cool artists and DJs? So there's a photograph I took of one of my favorite DJs. This is MCs. This is Q-Tip from a tribe called Quest, and this is Questlove from the Roots crew. And so, so we thought, wouldn't it be Alex? You stay here.
Oh, okay.
You stay here. You're stay here with me. What would we do if we could make music from some really cool artists? And so, so I had the good fortune a couple of months ago. I was giving this talk in the same session as this guy, DJ Jazzy Jeff, and so I kind of cornered him in like the dressing room, the green room of our area where we were giving a talk, and I kind of set up a little DIY lab, and it was all very coordinated with Alex, and she prepped all this stuff. And so basically, I was explaining to Jeff this whole project, and Jeff was so cool. He was just instantly like down. I was like, hey, dude, is it cool if I like get some of your bacteria and we make music from it? He hadn't heard anything yet or even seen the talk, and he was like, yeah, man, this sounds awesome. I'm totally into it. And so, so Jeff was like, hell yeah, let's do this. And so he started, kind of, giving us some of his oral microbiome. And, and this is my favorite picture of all time. This is DJ Jazzy Jeff inoculating LB, like his own like bacteria onto an LB plate. And look at how happy he is. He's like so gloriously happy to be a biohacker. And so basically now he got so into this project, we're collaborating with him to do a secret track on his new album. And he's been like telling his other DJ friends, and so we're kind of I don't know where it's all gonna go, but Alex is gonna take us to some awesome places, and we're gonna make some really cool music. We were working with Coco Roberts, who like previously dated Drake. This is me explaining to Coco oral sterile technique, and basically we're working with kids, we're working with some musicians, and This project has been a lot of fun. George, of course, has given us some of his bacteria, so we're making some music from that. And yeah, so the final thing that we're doing, so this is the random fun fact about me, but I'm the official iGEM DJ, and so I've been doing this for the past couple years. And so this year for iGEM, I don't know if Kim Demora is here, I know Thomas is here, but we're doing something a little crazy. So we're gonna do a project where we're gonna try to make music from the collective microbiome of the entire iGEM competition. So we're gonna get every single team from around the world to give us these plates, and like iGEM is letting us use some like high throughput imaging systems to actually try to get all this data. And so we're gonna try to make that happen this fall. And so yeah, so Alex is basically leading all of this work, and so a big round of applause for Alex, everybody. Thank you, guys.
Okay, I'm going to try again now. Thank you so much, Alex and David, for showing this awesome DJ booth. Okay, so I forgot before the panel started to actually introduce the idea of bioart and biodesign and why it's so important to have. I can say that coming from an arts and design background, that having bioart really opened up my mind to what I can do as a designer, and also opened up a whole bunch of questions about the ethical, social, and implications of what it is possible to be. As we know, there are brilliant scientists everywhere here trying to make the world a better place, and I think it is the role of bioart and design to ask questions like, what is better? Like, what is better for you or me, or a woman, a transgendered man, or someone with a prosthetic? So I think that bioart and design asks these questions and make them really emotionally tangible and understandable from everyone from in academia to like a 13-year-old girl on YouTube. So that being said, it is my pleasure to introduce to you Mary Magic. She is my lab mate in the design fiction group, taught me so much about bio, completely inspired me, and actually transformed my notion of what it means to be a woman and what gender is in general. So as she's an award-winning artist, she was featured at Ars Electronica recently. So without further ado, I'd like to introduce Mary to the stage.
Wait, is there
I don't know where the clicker is. David, is there a clicker?
Hi, everyone. So thank you for the introduction. I did get honorary mention at Ars Electronica this year, and I thought it would be fitting to start with this slide that's actually critiquing the festival. So last year, an artist named Heather Dewey Hagborg, who's a personal hero of mine, she started this campaign called #KissMyArs. And basically, she's critiquing how traditionally they always award the highest prize to men. And so the festival only recognizes the narratives, aesthetics, voices of men in general. I like this campaign. I think that it's trying to point to a really systemic crisis in media arts. But I don't think that the problem is just that we need more women. I think that we need a plurality of voices. So, yeah, and the festival was trying much harder this year because they helped to organize a panel called Feminist Climate Change. And during this panel, I got to hear amazing theorist Alla Mitrovanova from Russia. And she states, feminism is not just a way of thinking for women, it is the core of a new ontology, a new way of thinking, a new methodology that you can put into practice, that you can use to address these larger systemic issues. Donna Haraway, in her latest book, Staying with the Trouble, she states that, well, in order to tackle these huge problems that we face in Anthropocene, like ecological devastations, etc., we need to think about the tentacular connections that we have between non-human species, between our shared environments. And it matters what stories we tell. It matters what stories tell stories. It matters what thoughts think thoughts. It matters what worlds world worlds. That's a quote from her, one of her speeches on this book. So, narratives matter. Similarly, a theorist named Karen Barad, she talks about these interactions. Well, going back to these tentacular connections, what happens when laboratory science decides to comprehend, try to comprehend what's going on in these web of deeply connected interactions that we live in? Well, by definition, laboratory science is trying to understand phenomena, right? So we use these apparatuses to basically make cuts in this huge web of interactions. But when we do that, it inherently skews what we see and what we don't see. So basically what she's trying to say is that laboratory science is inherently biased. It's inherently exclusionary. And this skews our matter-making practice, our material discursive matter-making mattering. So to give an example, I research a lot about hormones, but more importantly, I'm interested in how hormones are used to define bodies, how to define male and female binaries. This is a very old photo, and basically when they were trying to identify and isolate hormones back in the early 1900s, they had no way of visualizing whether they had successfully isolated estrogen or successfully isolated testosterone. They had to do these very early bioassays where they would remove the testicles of an animal, then administer it testosterone, then record what they observed. And then the scientists would say, well, the animal became more territorial, more aggressive, so we think that, yes, we have successfully gotten testosterone. So you can see that in this experiment, they're very, very biased in the way that the biases they have about gender, about masculinity, already comes into the research. This is a synthetic estrogen called DES, and it actually came from the petrochemical industry. But then they started to observe that it would have these feminine effects on whatever animal was being exposed to it. And so they started to prescribe this to women who were too masculine, women who were too tall, women who were going through menopausal symptoms, who were losing their feminine essence. So I want to go through a couple of works that really inspire me, works that really try to decolonize what we perceive as male and female. So this is an artist named Fanny Sosa. She's really amazing. She's famous for giving these twerk shops or twerking workshops. But for her, twerking is a way of remembering, it's a way of resistance. And she's often approached by people who say, well, if I'm white, am I allowed to twerk? And she's like, yes, you can twerk if you're white, but just know the history. So she wrote this really great article called Biohack is Black, and it's one of my favorite articles I've ever read. And she wrote it in a response to an article that came out titled, "Cyborgs are all among us, they're called women," because women with IUDs and these implanted hormones are considered cyborgs. They're considered almost transhuman because they have this ability to regulate their reproduction. And she said, well, no, that's very problematic to say that because women were always in charge of their reproduction way before these pharmaceutical industries came in and basically told them, hey, take the pill, this is gonna help you control your reproduction. her article, Biohack is Black, was actually published in the 3D Additivist Cookbook. And this is also a really amazing curatorial project by Alison Morrison and Daniel, sorry, I'm saying all the names wrong. But basically, this is kind of supposed to be about, inspired by the Anarchist Cookbook, but not so much. It contains all these recipes and writings of artists and designers who are trying to reimagine our future, a future that is right now being taken over by technology, 3D printing, things like that. And they say that the feminism we're interested in is a philosophy of more than just women. It is a philosophy of non-male, non-cis, non-white. Challenging dominant narratives is crucial to maintaining plurality. So one of my dear friends, Clau Kinky, she has been doing this project for some years now called the Anarchic Land. And basically, the gland that is in charge of the female orgasm is currently named after a man. And this is something that is a form of erasure, because the 3 women who were experimented on for the advancement of modern gynecology, Anarcha, Lucy, and Betsy, they were Black slaves living in Alabama, and nobody knows their trauma, their names, their experiences. And so she's trying to bring that narrative back into the way that bodies are named and defined and labeled. So if you ever see a picture of the gland in the female anatomy and it says the Skene gland, please correct that person. It's actually called the Anarcha gland. So another dear friend of mine, Paula Pin and Gaia Leandra, they've been collaborating together on several occasions on making DIY tools for self-gynecology. So building webcam microscopes that are attached to speculums, 3D printed speculums. And this is a way to understand your own body and gain more autonomy over that. Another good friend of mine, Isabel Burr-Ratty, she is a filmmaker and a performance artist. And she's interested in the ontological crack between the native and the engineered. And I really love that, that dichotomy. So she goes around with this beauty kit, and in this beauty kit contains all these like facial moisturizers and pills that are made from the products of the female reproductive system. So she has pills that are made from the dried blood of her menstruation. In this picture, she's actually making Bloody Marys from these pills. I think Eric Zepka had a taste of that. Yeah. And yeah, she has this, this G-spot orgasm spray and all kinds of stuff. So my own personal work, I mentioned previously, I'm interested in how gender is codified by hormones. So how hormones like estrogen and testosterone can contain this biopolitical influence over our bodies. So for the past 1.5 years, I've been generating these DIY protocols for hacking estrogen. So for example, I have these mobile labs for detecting and extracting estrogen from fluids like urine and waterways. And this is a way for us to see that the estrogen is all around us, that we are completely colonized by this molecule. It's existing with biopolitical agency. And I've also created a fictional cooking show. It's a very short film called Housewives Making Drugs. And it's featuring 2 trans artists. And basically, they are sharing estrogen recipes with each other and their audience. So aside from how hormones are right now regulating and controlling our bodies, hormones are actually also polluting our bodies. So as a product of petrochemical, pharmaceutical, and agricultural industries, these hormones are all around us, these molecules that are hormonally active. And you've probably heard of the media saying, oh my God, there's trans fish and trans frogs in the rivers, and it's because of women taking birth control pills and peeing that into the water. Well, that's kind of true, not really. Well, to call them trans is the wrong terminology. I think intersex is the better one, but yes, it's true. A lot of species have been mutating because of these hormonally active compounds, these endocrine disruptors. And humans are not left out in that. We're also mutating. And I kind of see this as a collective mutagenesis, kind of like embracing the horror together. So I want us to not get too caught up in this fear. I mean, it's really, a lot of people come up to me and they're just very shocked when they hear about endocrine disruption. But my project's not really about scaring people. It's about how to neutralize that fear, how to come up with new narratives for us to refigure this toxic world that we live in. So what about queer body worship? What does that mean? This paper came out in 2016 where scientists hybridized 2 species of fish, and the result was a hermaphrodite species. And then they called it a hopeful monster. And I think this is a really beautiful terminology to use, because why can't our queering bodies also be considered hopeful? Because in a way, you know, so many molecules are interacting in our body on a daily basis. I mean, millions, we can't chart all of them. And you know, we can think of our bodies as undergoing some kind of glitch, right? We're all glitch art in a way on the molecular level. So yesterday, Stephanie Wischitz, she's kind of summarized 3 categories of feminisms. And I would say that I align most with xenofeminism. And to end this talk, I would like to just reiterate, if nature is unjust, then change nature. Thank you.
Thank you so much, Mary. So next, I'm introducing Adam Zaretsky, who I had the strange and wonderful experience of meeting here at MIT Media Lab in the kitchen. And because I'm having such stage anxiety today, I'm actually going to just read his credentials so I don't miss anything. Okay. Zaretsky is a wet lab art practitioner mixing ecology, biotechnology, non-human relations, body performance, and gastronomy. Zaretsky stages lively, hands-on bioart production labs based on topics such as foreign species invasion, pure/impure, radical food science, edible/inedible, jazz bioinformatics, code/flesh, tissue culture, undead/semi-alive, transgenic design issues, traits/desires, interactive ethology, person/machine/non-human, and physiology, performance/stress. Former researcher at the MIT Department of Biology for the past decade, Zaretsky has been teaching experimental bioart at San Francisco State University, Symbiotica, Rensselaer, University of Leiden, and his own school, VASTL, the Vivo Arts School of Transgenic Aesthetics. Okay, I'd like to welcome to the stage, Adam.
Thank you so much, that was really long. I get the clicker. Okay, hi, I made it. Last night I was in Washington presenting this one, so I changed the name in Washington from intentional genetic modification orgiastics to this, but don't take it too seriously. What I want to talk about, hold on a second, we should go back. Can we play videos? Yes. Try and play a video. We got to start with some music. Good morning. Crank it, yeah. I'll just try to do air guitar for you. I missed yoga. I like showing videos of myself because I worked with Robert Arneson, and he's into like self-portrait as, oh, making fun of yourself. I had the clearance to use this plasmid, which was known as lunatic fringe. I like lunatic fringe. I had a choice between radical fringe and lunatic fringe. I like the song. Lunatic fringe, I know you're out there. I know you're out there. And you can order them. There's a shopping cart, and it's $60, you know, or $55, and you get, you know, a couple of microliters and, you know, like 20 doses, etc. And that's where the information flow returns to the life world. Is you buy it, you shoot it in, or you inject it in, or you electroporate it back into nuclei, and you hope they then take your little infectious donut into their heritage. Lunatic fringe, as a plasmid, helps or disrupts or enhances the body plan. So there we go. We're helping or enhancing or just disrupting and mutating. It's hard to know which we're doing. We actually don't know what we're doing. And I've got about 10 minutes to sort of talk you through my program here. Let's see. I'm going to tell you a little bit about BioArt and human gene editing, but really what I want to talk about is the making of transgenic humans, is the making of genetically modified humans an art form? Like, in other words, is it a new kind of sculpture? First, I'll talk about BioArt a little bit. This is a DNA lab I did down in Mexico City. We made DNA from all these samples at once. So that's hybrid DNA. It's kind of a mash-up. I developed a lot of this working across the street there for a couple of years as a researcher. I'm interested in ecology and food and the non-human and the body arts like performance arts as a way of introducing what BioArt actually is, because anything that deals with life in art deals with some of the same issues, which is like, are you okay? Is your art okay? Do you care? Stuff like that. So it can be as simple as an eco-art lab in the streets where you're throwing seeds in soil, but it can get more complicated down here with tissue culture art and molecular biology art and mutagenic developmental biology art, which is like making transgenic organisms as art. I do a lot of this like DNA isolation in the street with people. It's very fun. You know, really messy, super, what is it, exorbitant, baroque, whatever you want to call it. But I want to talk to you a little bit about transgenic hereditary biology arts. It started with physical manipulation of embryos. It then led to adding poisons to them as they were growing for environmental toxicology. And actually, transgenics is both physical and poisonous together. In other words, it's a subset of both. This is standard developmental biology, embryology. So, I mean, just keep it in mind that it's physical and it is detrimental for the most part. Every day there's new news. Like I was like, oh my God, Frederick Lanner from Karolinska. I knew they were ahead. They're actually editing healthy human embryos instead of ones that are gonna die, you know, automatically, like trisomy or something. So this is cool, except for like yesterday, it turned out that Kathy Nyakin got the award for like waiting a couple of years and got a license to also knock out genes from healthy human embryos. So I'm just trying to say this is going on, like this sort of, what do they call it? Oh. I pressed that instead of laser. There we go, good. This sort of cut and paste stuff, this kind of like people that are into editing audio or editing video, it's sort of like one from column A, one from column B, cut and paste stuff is going on inside of our collective genome. Okay, I call this transgenic hereditary cascade embryological developmental biology arts. There are also these other names for it. I like to think of myself as a sociopathic stillborn artist, but that's a different story, okay? So I'm looking here at like George Church's how to infect the human genome with CRISPR, and some of this standard stuff, which is like the bioethics of it, right? And I'm looking at what's interesting right now is they're starting to realize that if you add the CRISPR construct with the sperm at the time of fertilization, there's less mosaicism and supposedly less off-target mutations, like less cancer caused by this incredibly good targeting system. The place that I'm looking at is it actually migrates from the hindgut. This is a cross-section of a human embryo, like a chop. And that's our tail. You remember your tail? It went in as your genitals came out. It's a long story. They migrate. The primordial germ cells migrate here to that groove and make the oogonia and the spermatogonia that go to make your sperm and make your eggs. When you inject a construct, a genetic modification with the sperm, that affects not just the cells of that organism, but also the cells of, what do you call it? The hereditary cascade of that organism, right? Their kids and their grandkids throughout all living time, which we don't know how long that will last. But I consider it not just to be art, but also a signature, maybe just graffiti, right? Like tagging the human genome. A thumbprint, I was here, you know? It's maybe writing on the bathroom wall of the human genome. Permanent narcissism, that's sort of business as usual right now in the US, so I'm not even gonna touch on that. So here are some of my people that I think, maybe we could do this. MIT Media Lab, do you wanna give prizes to the sculptors of the year for sculpting the human genome? These 4 deserve prizes as not just sculptors, but new media producers. What is it? It's new media? This is like art speak. Huh. I don't know. Time-based new media? What is it when you alter the human genome if it's just art? I went to the Human Gene Editing Initiative down in Washington, and I was like invited guest from David Baltimore. He ran Asilomar. I called him up. I was like, I want to come as an artist. And they had me, you know, they put up with me. And I'll show you a little clip from that. We might have to play this video. That's Hank Greeley from Stanford and Jennifer Doudna. I don't know. She's like the CRISPR inventor from Berkeley. Let's play the videotape. Who's on it? I'm here with you.
Adam.
Adam Zaretsky, bioartist, and I identify as queer like us. I wanted to just talk about a little bit of a fear of human, we're actually thinking of programming our own future genome, and the concept of the human is less queer than the technology. This is where the artists sometimes come in. I'm looking at health or health betterment, which is optimization. Enhancement falls under optimization of the human genome, but that would be us making ourselves into a monoculture based on traditional humans. It's really strange. We would be heirloom GMO humans or organic GMO humans without looking at alternative aesthetics. We are falling into this sort of the problem of disability studies for transgenic humans. The psychosocial problems of transgenic humans might be, some of it is because anything but a traditional human form is perceived of as monstrous, and yet we're actually altering form to a great degree, and it's under the aegis of health itself, and that's not enough.
Responses, thoughts? I mean, I do think that civilization, or whatever you call what humans have been doing for the last 10,000 years, has made huge changes in who we are, what we look like, and what our allele frequencies are. So there is some continuity, at least, in our discontinuity.
Thank you, Hank. At least he answered. Okay, so what am I trying to do? I'm trying to actually ask, I can't believe this guy's so smart. I smoke a lot of pot, and I'm like, I said that? That's pretty good. I'm just saying, like, why are we sticking with human or superhuman? Why aren't we doing different things? What I want to do, though, is spell out a little bit of, like, the architecture of where we're talking about and what we're talking about. And I'm talking about this in terms of intentional genetic modification orgy, right? Like, not just like, it's for curing birth defects. That's the reason to get the research through, but really, or for NASA or the military like DARPA. But no, just to like go balls to the wall with the technology, right? So to say. So one thing is, where is this happening, right? And this is like, I can't cover the list because I've got 5 more minutes tops, but You can get human embryonic stem cells mail order from like UPS or FedEx will ship them, right? There's plenty of extra embryos laying around. You can actually make your own embryos. There's human embryonic stem cells in orbit around us right now on the International Space Station. Okay, so these are just places where this might be happening. Then there are places where it happens on your body, right? If you're going to become a provider of genetically modified children, it could be just your body and your ovaries or testicles become affected. It could be your sperm that you donated 10 years ago, earlier, less, right? Today. It could be tissue or organ cultured ovaries or testicles that are milked for years after you're dead. Okay, there's a lot of places on your body that you can provide future human cell sculptures for. And then they get implanted, right? You can probably just buy a bunch of pre-implantation embryos and jam something in there. I mean, into the embryo and then implant it, right? And I'm talking about like, I'm forming a corporation, I want to make tribes. Like I'll sell you 1,000 kids, right? They'll be ready in 18 years. Put your money down. They'll have dolphin fins on their head or something. There are different ways of getting the genes in, and I'm kind of obsessed with this. In fact, I'm going to Moscow in January to do 3 labs. It's called Methods of Transgenesis, Shoot, Shock, and Inject. It's kind of like, you can shoot them with biolistic guns, you can inject them with microinjectors, you can shock them with electroporation. And then there's other stuff like liposuction. And I'll show you a few labs that I've done like this. One was micro-injecting. I made micro sushi here at the Media Lab with Mary like last year. But micro-injecting zebrafish embryos with algae to try to make them solar powered at like a clear channel event in the Netherlands. Wow. You know, just using a little microinjector and an Intel microscope and some fish eggs. Isolating DNA from a centipede and electroporating it into my sperm, right? That was for Jazzy and Leonardo. Thank you, MIT. Here's like a cartoon of how I did it, like a do-it-yourself centipede sperm lab. Zap, right? Or mixing shit, blood, sperm, micro, like nanoparticles of gold and DNA with my own. This is a gene gun. I just used metal petri dishes, a sledgehammer, and a jackhammer. It worked. It's amazing. And I painted with it. It's an art media production lab. So I like sort of set up this printing lab. We printed it on canvas and on each other's forehead. It was like a lot of fun. Or recently I did this, a DNA coconut liposuction using DNA from the garden, like giant lettuces and kales, and we kind of mixed it with extremely low pH coconut butter and made like an 8-person massage table and then gave people liposuction massages. You know, you like that? Yeah, it was pretty cool. So I'll try to wrap it up with just talking about aesthetics because I've touched on it, but this is really kind of important. Aesthetics is not just what makes you feel good. Aesthetics is the range of possible feelings that you can feel due to ecstatic art contact or strange circumstances, right? I would say the aesthetics of redesigning life forms, a lot of times, for me, it's important that it's like the widest range of feeling should be expressed anatomically. Like, you know, bittersweet, demure, sultry, yet despicable, right? I chase that one a lot. You know that, David. Yeah. Anyway, so this is what I'm sort of up against. This is the reason why you're supposed to engineer the human genome, right? To make us stronger, more intelligent, able to leap tall buildings in a single bound, to make us better warriors to fight, you know, whatever other nations have also the same programs of making transgenic warriors, and also better workers, perfect slaves. The pornographic potentials, I'm ready to get in on that one, but you know, and if we got totally pure, that we would be some kind of Euclidean solid or or a perfect platonic sphere with 0 orifices and no membrane transport. We could just think. Other options. One, we could just totally mess with our body plan, right? Like, get 20 toes, mess with fate maps, hawks, packs, like these so-called master and mistress genes, ectopic eyes on the back of your knees if you're kind of into emo, right? So, other things we could do is just mix the entire life world. We've got these bioinformatics databases that are untouched. The human genome is open. What's wrong with putting a platypus medulla on the back of your kid's medulla? It's got a double medulla, one's marsupial. Is that enhancement? What about upping the cannabinoid pathways so that you can like squeeze your kid's glands and sell it as like a, you know, mannitol replacement? I don't know. Look, so there's like beyond cut and paste Photoshop stuff and beyond kind of, what do you call it? That sort of jellyfish human. There is also like thinking about space exploration, right? If we want to get exobiological, how do we do it? We might have to sporulate, right? And send those spores mixed with sand into space and see where they land. And maybe all those mushrooms will be shaped like Arnold Schwarzenegger and it'll be life as usual again. Just intergalactic. So I came up with this. It's a roster of all the mutant design. You know, I mean, like, it's not complete, but there's skunk and poodle and dolphin brain chunks in your brain so you can MRI yourself and design chocolate chip cookies from that. And then, like, you know, like a gecko person, so we don't have to do all that 3D rendering. I am worried about psychosocial problems for transgenic humans, but it's the same as problems for anybody who suffers from otherness, difference, queerness, even just lapses of human behavioral tropes, you know, like if you freak out. So, I mean, I'm worried about it. At the same time, I'm amazed. And do I have time for one more 1-minute video or not really? We're gonna cut it there. I made some plans. We'll get into it. I brought some writings if you want some. I love you all.
So thank you so much, Adam. So next I have the pleasure of introducing Cosima and Daniel. So Cosima I don't have the pleasure of knowing yet, but has a fascinating Google search results as the real Cosima of Orphan Black. As a science consultant in Hollywood, her official job is to use her PhD in the history of philosophy of science, technology, and medicine to make TV and movies more believable and thoughtful. Daniel is a director and producer who owns a company called Loud Roar Productions. His latest documentary is Ghost of Our Forest, which chronicles the struggle of a displaced indigenous tribe in Uganda. So welcome.
Let's keep it going again for Daniel and Cosima. Yeah. Woo! So we're just gonna do a quick little, 5 to 10 minute interview with both Daniel and Cosima. So you may have noticed that in addition to MIT Video, there's also been kind of another camera crew that's been kind of roaming around and following us. And so that's basically been both Cosima and Daniel's project. And so, yeah, it's been such a really interesting experience getting to know both of you. And maybe you can both just briefly introduce yourselves to the audience and say a little bit more about why you're here. So.
Hi everyone, my name is Daniel, as you already know. I am from Toronto, Canada, and I'm very excited to be here. And part of the reason why I'm so excited to be here is because I'm a film director with no formal scientific training. I've been saying this all day yesterday. I'm the kid who failed grade 10 science. And so learning about this world and some of the issues that are being discussed here has been very empowering for me because I get it. And I'm like understanding these concepts that I once thought were reserved for like smart people at MIT. But now I'm a person at MIT kind of, and so that's really cool. So it's just really great to be here and to be having so many wonderful conversations and to be getting to know so many of you, and that's been really wonderful for me.
Actually, you guys all don't know, but as part of the conference swag, you all get MIT honorary degrees. They get conferred. It just happens automatically, so you don't even have to do anything. Trust me.
That's excellent, because I actually got kicked out of my PhD program, so I'm looking for one. I did not finish my PhD. I think that that was part of my introduction. I actually left my PhD program in the history and philosophy of science, technology, and medicine to work in television as a consultant, primarily motivated by the fact that I didn't have any money and I needed a job and I couldn't afford to continue with my PhD. I, like Daniel, am also Canadian, and hey, fellow Canuck out there, somebody like that. And we will tell you a little bit more about what our project and our documentary is about, but I wanted to tell you a little bit first about how I became interested in citizen science and DIY biology and biohacking. I've long been interested in how science, and biology in particular, has been marshaled into the service of politics since there was a discipline called biology. We're talking about something called life and bodies, and it cannot but be political. I'm really interested in the idea of hacking as a form of intervention and DIY science as a form of activism and protest, and how important it is that people have the agency, not just only of your own bodies, of our own bodies, but of the access to knowledge and the ability to have conversations. And one of the things about academia that caused me a lot of issues while I was there, I aspire to be a good scholar, but I'm a terrible academic, is because I feel like there are more effective and more broad ways to actually have conversations in the world about the issues. I have no capacity or desire to try to answer the hard questions. I'm really interested in problematizing our assumptions. And I became really influenced about biohacking by a few people, two of which you've heard speak today. One was Mary Magic and another is Adam Zaretsky. Mary and her colleague Ben, they did a series called Dissect, a series of short videos, and I use those when I consult with writers who work in the biological sci-fi realm. And Adam, which is just a never-ending source of inspiration and wonder. So that's kind of the short of how I end up here.
So really briefly too, so again, I think we have a lot of, I think all of us actually are all nerds in this room, and so how many of you has seen the show Orphan Black or are fans of the show? Sorry, so we have quite a few folks here, yeah. And so this is Cosima from Orphan Black. This is actually Cosima. So I don't know if just really quickly you want to just say or explain how that all happened in your kind of Cosima-ness into TV.
Okay. Again, like I said, I was working on my PhD. Well, I was working on a master's when the idea for Orphan Black began. It was an old friend of mine named Graham Manson, who was one of the co-creators. It took him a long time for anybody to want to buy Orphan Black. Nobody thought we could pull it off. We always called it the weird little show, the little engine that could. We're here with another one of my colleagues, which is Mackenzie, standing up. Perfect timing. Mackenzie, I've also had the great honor of working on Orphan Black since its inception. She's one of our producers, and she's also one of our producers for this documentary. And Graham used to call me in the middle of the night going, "What do you know about clones?" And I'd be like, "What kind of clones? Like human clones, potatoes clone themselves, cells clone themselves. Like, what do you want to know about clones?" It became a really excellent way to have a conversation about different kinds of issues. We use science as a buttress for other kinds of conversations that I want to have. Orphan Black is a feminist show. It's a science fiction, but it is decidedly about a platform to talk about other issues that I think affect everybody on the planet. So that's kind of how I came to be. I didn't name the character Cosima, by the way. That was Graham.
So again, I think you had this amazing and really successful show on TV, and now you and Daniel are working together on this amazing new project. And getting to know both of you and Daniel, I think, as you mentioned before, this is kind of one of your first forays into science. I'm curious, if one, just explain a little bit more about the project, and then two, how do you view kind of documentaries and science and kind of telling this story and you know, how does that platform, do you think, kind of enable you to communicate some of the issues that's facing this community?
Well, first and foremost, are we on here? Here we go. First and foremost, so often when we think of science documentaries specifically, you know, maybe our minds think of like being in science class and watching these very sort of antiquated and boring talking head films of people explaining concepts and things like this. And one thing that was very important to me when I was approached to work on this really cool story and this cool project is to make sure that like this film is as like kick-ass and sort of like punk rock and diverse as this movement is, and to not feel like sort of a typical science documentary. And that's something that's going to be very, very easy to accomplish just because so many of the folks we're interested in collaborating with and engaging with are just so unique and just amazing. In terms of what the project is actually about, I think Cosima can really speak to that.
So what we're trying to explore, again, is citizen science. And one of the things I'm really interested in is this relationship between those who work in license-regulated labs and those who don't. I mean, there's a lot of questions that can be asked, but in my mind, one thing that is really interesting is if it's only those. Look, I'm really interested in questioning authority. So we defer to people in white lab coats. I'm absolutely all for science, and I'm absolutely for the authority of expertise. But when there are those who are behind the hermetically sealed doors, can only do the certain kinds of science that we're talking about, they're also the only ones who can decide how it gets to be used. And I find that concerning. So I want to interrogate that and what does that mean? I don't have an answer for it. I also became really frustrated when I started looking at these issues of DIY biology that almost across the board, 95% of what I saw by most media was biohackers and bioartists as some kind of fucking terrorists. And I thought that was really, pardon my language, sorry, I'm a terrible swearer, sorry. I found that so shallow and tropish. It's not my experience with people. It doesn't reflect any of the conversations that I had. And you know, there's a lot of bizarre things that are going on behind closed doors too. So I'm not here to make anything sinister. I'm here to understand what this relationship is and what this has to do with civilian agency. It's not just the human body I'm interested in, it's every living thing on the planet that is in rapid change. And we need to equip ourselves with knowledge. And I really believe that citizen science is one of the ways that that conversation and that kind of agency can be harnessed and endowed. So that's what we're looking at.
Amazing, amazing. I mean, I think you summarized so well a lot of the themes of this conference and kind of why we're all here together is thinking about, you know, how does biotechnology move beyond kind of a lot of the traditional institutions and become more of a grassroots thing. And so, you know, just to kind of wrap up then, you know, you've been here for a day, you know, we've got a day today and a day tomorrow. You know, can you talk a little bit more about, you know, your intentions and kind of your hopes in interacting with our community here over the next 48 hours or so? Yeah,
sure. I know from my perspective, what I'm really interested in doing is just meeting everyone and just hearing about all the cool projects that are taking place, where they're taking place around the world, and just what problems they're trying to solve and what real-world issues are being addressed through this, you know, do-it-yourself hacker ethos. And this is what I'm really interested in. And so if anyone has any cool ideas or projects or things that they're going to be working on within the next year, I really want to know about them and hear a little bit more about what's going on and things like this, because there's a lot of room for collaboration. And I think we're approaching this film with the same sense of collaboration that this whole gathering is in the spirit of. And so we have a few folks here, and come find us and come chat with us, and we'd really like to have a conversation.
Yeah, it's about the conversation. There are some people that we have reached out to, some people I have met before that have really been so generous in giving suggestions and guiding us to, like, you should look at this person, you should look at these projects, but there's so much else that's going on all over the world. I don't know what it all is, so this is also exploratory to meet people, to get your opinions, and to let you know that, you know, this project is probably will take us about 1.5 years. We're looking at it probably being aired in the spring of 2019. We've got a lot of work to go, and we will be all over the world. This is not just North American or Eurocentric, and that's also really important to us. So I want to be able to talk to people and actually have your voice. I'm not here to label for you what you do or what you are, but it is undeniable that there is a phenomenon of DIY science, and it makes me feel hope. So that's basically what we're here to explore. So please, we're really friendly, and please come and talk to us if you think there are things that we should be concerned about, or that you're concerned about, or that there's something that you know that you're like, you should check this out, and it's off your radar, and if it's not on your radar, you will be remiss. So we really, really want that input. It's really important to us to be able to do it right and as best we can. So.
Well, yeah. So anyways, Cosima, Daniel, it's been so awesome. I mean, Cosima, we hung out in Switzerland and had a really great time, and Daniel, it's been awesome getting to know you here. But everybody, please meet them both. They're both really, really wonderful. And again, if I understand correctly, this event is kind of the start of this whole project.
Yeah, this is the inaugural. This is the inaugural filming.
This is so. You know, hopefully, you know, some of the characters in your film will be really kind of started, and their journeys will start from this room right here. So, thank you both so much again for being here, for sharing your stories, and we look forward to continuing hanging out with you both over the course of the weekend. So, another round of applause, please, for Daniel and Cosima.
Thanks.
Oh, you can leave them under there. And another round of applause for Annie for doing such a great job hosting. Yay, Annie.
So thanks so much again. So next I have the pleasure of introducing Orkin Telhen, who I have the joy of calling my friend, one of the most brilliant, kindest, creative people I know. He is an associate professor of fine arts at the University of Pennsylvania and the co-founder of neutralize, a company specialized in designing new technologies for biological design and synthetic biology. He holds a PhD from right here at MIT from the design computation group, and has been shown internationally, including Istanbul Biennial, the Armory, Ars Electronica, all kinds of amazing places. So without further ado, here's Orkin.
It's a pleasure to be on the stage again in an hour. Today I'm going to talk not about my own work, but I'm going to talk about a little bit how do we extend these efforts of DIY biology and citizen science to different parts, different disciplines, different communities all around the world, art communities, design communities. And I'm also especially thankful for this session because we heard very different voices in the art, from the art world, from the film world. So the voices, we need more diversity in these voices to really bring DIY biology into people's minds so that we can really think together in larger issues. So I am a designer and an artist by training, and I've been teaching synthetic biology and biological design for a while. And in this crowd, biological design is something roughly we know. You know, we use organisms to create new kinds of products, and those products could be anything from food to cosmetics to biomaterials and so on. So that is happening. You know, this is a bunch of things that are already in the market or some research that is really well known. But we really don't know what comes after this, or how do we arrive to these things. So I've been teaching synthetic biology for 5 years, and I'll show you some slides from my student work. And we work on mainstream biological design. How do you really work with organisms to make very practical things? You know, like, can you really make biomaterials? Can you really work with food in different ways? But we realized that, or I realized after 4 or 5 years, that there's a little bottleneck. I mean, you have seen many slides in the past 2 days and the Hello World slides. We kind of look at the same things. The kind of activities that are happening in the community labs, biolabs are kind of similar. There's a lot of incentives to learn biology, but we really are having a bottleneck in terms of coming up with creative and critical applications. So how do we diversify that? So my role as an educator, I want to give back a little bit to the community, like a little mirror of what kind of things that are out there that we have developed, not as commercial products, but good cases. Good cases that can be documented, that can be really very well articulated, not necessarily on the biology side, but also on the design side, so that we can really improve these best practices for the new students, new people who are joining these bio communities, as a new ways to really think with biology without just being confined to, hacking certain things or developing new tools and so on. So diversify the examples. So a couple of examples from this initiative. So we decided to get this URL, design.bio. It sounded sexy and cool. And I started putting student projects out there so that we can document the process of how do we really, capture these best practices. Because it's not like Instructables, you hack together a couple of things and really make interesting applications. But how do we really, show off some good design? And this good design could be really materials that are really developed and applied and ready to be really turned into furniture or architectural components, or going back to, or sensors that actually work to really detect certain kind of things, so that you can take these examples and take to the next level. So it's also a shout out, after this brief introduction, we will have a 40-minute breakout session with Nuria so that we can really discuss these things. So it's not about showing what's happening out there, but also getting your feedback about, well, how do we grow this community of artists and designers by showing the kind of work we do to each other, as opposed to really being confined into community labs and different kind of experiences. How do we really, get heard, but also bring more people into the space so that we exchange, new ways of thinking. Now I will go very quickly because I introduced a lot of these concepts before. So there's a lot of artists and designers who want to get into the space, so they lack the knowledge and the tools. So we are working on that and different initiatives, but they also don't know what they can do with it. So that's a very important bottleneck. So knowledge is important, but what do you do with the knowledge is much more important after that, ethically, responsibly, and even also commercially. Then the other part of it is how do we, what happens after the workshop? I mean, all these community biolabs, you guys are doing amazing workshops, but what happens after that? When the students go back, do they come back again, or what do they come back again for? So these kind of questions are important, and I'm also throwing them back to you. So let's think them together in our breakout session. So one example of how this can be done, let me walk through this quickly. So we work with high school students, we work with BioBuilder. We are interested in really the future of food, in terms of how do we really bring biological concepts into food fermentation and food production, but now include designers into the mix so that we don't necessarily just focus on the biology, but take as a production of a food artifact. Let's look at baking, let's look at donuts as a way of thinking through all these issues as an integrated design exercise. So if you are an architect, Rhino, 3D printing and modeling, you don't really care about biology that much, but can we intersect our paths with biology through finding a common language? My timer says done, so I'm going to wrap up. But the important thing is, so if you think of food as almost like an architectural program, can you design the form, can you design the shape, can you just think about the ingredients that will go inside that food, so that you not only necessarily design the form based on how it should look like, but based on how much, how the yeast that's inside the dough of a donut, how much vitamin A it can produce, so that you can actually control the functionality of the food based on its form factor. So if you make internally more space inside the donut based on the form that you create, it will produce more carbon dioxide, it will produce more vitamin A, and you can actually have both a function and actually a chemical function and an architectural function at the same time. And then what do you do with this? Well, you document it really well, and then you write about this so that other people can make use of it as well. So here's a shoot out also to Biocoder, O'Reilly's newsletter. So we write it up and share it with a lot of people, but also it's a charge for all of us, the art and design community, to come up with these things and then write about them so that other people can systematically explore them. So it's not just about making the cool examples and showing them off, but also enabling other people to really work with them as well. So I'll stop here, and after the sessions ends, you can come with, Nuria and I will go in a special place, and we'll discuss with you guys what else can we do for design.bio and the design community in general. Thank you.
Thank you so much, Orkin. So next, I have the pleasure of introducing Allison. Allison is the program manager for the Bio Design Challenge, a university competition that pairs artists, designers, and scientists together to envision the future of biotech. Bio Design Challenge is a program of GenSpace, an awesome nonprofit community laboratory dedicated to promoting citizen science and access to biotech in New York. She has received the Hunt Fellowship for her work as a theater artist, creating performances centered on the emergence of new biotechnologies and their implications in the context of social and economic inequalities. So, without further ado. Thanks so much.
Thank you so much, Annie. It's awesome to be here today at this wonderful summit. So yes, as she mentioned, I am the program manager of the Bio Design Challenge. I am here today with my colleagues as well, Daniel Grushkin, who's the founder. You heard from him today talking about his work at GenSpace, and also Vina as well, who runs the program with us. So the Bio Design Challenge, as Annie mentioned, is a university competition that pairs art and design students with scientists and asks them to envision the future of biotechnology. It is GenSpace's largest flagship art and design program. So BDC's goals are threefold. One of them is to prepare young artists and designers to work with emerging biotechnologies. A second one is to build collaborations between artists, designers, and scientists. And the third one, which I think is really important, is to spark a public dialogue around these emerging biotechnologies. So this is a lovely photo. Orkan, he's been in lots of people's presentations today. It makes sense. He's fantastic. Basically, the Biodesign Challenge, we work with universities throughout the spring semester. We work with them to build their biodesign curriculum. And then throughout the semester, as they are working on their projects, we pair them with what we call expert consultants, which can be anyone from biologists to life cycle analysts to science historians to chemists. So it just depends on where students take their projects. So this is a photo of Orkan and Karen Hogan's class at UPenn. They've taken part in the BDC for 2 years now, in 2016 and 2017. This is one of my favorite photos that we've gotten from any classroom. So basically, yes, students work on their projects throughout the semester, and then at the end of the semester, one finalist team is chosen to go to the Biodesign Challenge Summit in New York City and present their project and potentially take home some prizes. So 2016 was our pilot year. We worked with 9 schools. And then in 2017, the program more than doubled to 22 classrooms, or 22 schools and 24 classrooms total globally in 7 different countries. And now for 2018, we are expanding the program to include 30 classrooms all throughout the world. So we're really excited. And then, yeah, so just letting you know, registration for that is open right now, and it closes on November 6th. So, to just quickly take you through a few of the projects, as you can see, we have our Biodesign Challenge little exhibition outside with some projects, but I would like to go through some of my favorites, and just, yeah, to just get started. So this first one is by some students from Fashion Institute of Technology in 2016. They were actually the winners of the Biodesign Challenge in 2016. They were looking at how polluting the fashion industry is and wanting to create a more eco-friendly biotextile. So they created this awesome algae biotextile that you can knit with and stitch with, and it's really fantastic. There are some samples out there. They actually have started a company now, which is really exciting. And something more exciting, last week we just heard that they were awarded the National Geographic award, they're like Chasing Genius award, which is really amazing. So congratulations to them. Yay! It's really fantastic that so many teams have continued with their projects and started companies. So please go out, talk to them, look at their work. They'd be happy to answer some questions. One other project Here's a short video, actually, of them making their algae material. So they basically extrude it into this calcium bath, as you can see them doing here. And then when they take it out, it's somewhat solid. And they're able to stitch with it, knit with it. I love this. So yes, please go out and talk to them. And this next project, which we also have out there, was a project from School of Visual Arts in 2016. And they were also looking at pollution from the animal farming. So they were looking at a future where we perhaps did not eat meat or did not produce meat. So they were looking at how to perhaps enrich the plants that we have. So they created this tomato called the Mayo tomato, which they jokingly called it the beefsteak tomato, because they actually went into the lab and put a gene for the myoglobin beef protein into the tomato and actually made a beefsteak tomato, which is amazing. These were a bunch of art and design students that worked with scientists. One of their mentors, Sebastian, is actually here today if you want to talk with him more about it. But that was really amazing and something that we did not expect for them to do, to actually get into the lab and make something, which was amazing. So yes, that is outside. Please go out and check it out. So yes, thank you so much for your time. As I mentioned, the Biodesign Challenge 2018 competition is open. If you would like more information, please feel free to contact us. And then also more student projects are on our website, so feel free to check us out. Thank you.
Thank you so much, Allison. And Allison and I and Lena are going to have a little fireside chat about biodesign and speculative design. So if you're more interested, please join us. Also, my curator is here and will kill me if I don't do this shameless plug. I have a show coming up in Boston on October 6th. It's called The Laboratory of Longing. So if you're around, please check it out. And without further ado, David has some microbial surprises for you, it looks like.
Sorry, guys, this is a little complicated objects. Okay. I got it. Okay. Okay, all right, so a couple of quick announcements. Okay, so we're going to do our first set of breakout sessions now, okay? And I'm going to explain how that's going to work. And this is relevant. Okay. So what's going to happen is we have 3 breakouts, okay? Orkan and Nuria are leading a session. Again, Annie and Lena and Allison are leading a session, and then is Adam Zaretsky still in the room? Adam is going to lead a session, all right? So this is how this is going to work, and this is how all of the breakout sessions and on-conferences are going to work, okay? So I'm holding in my hand Joey's beloved stuffed microbe collection, and they've been lovingly mounted onto these plastic poles, okay? And so what's going to happen is, for all of the breakout session leaders, you can come on stage now, actually. Just come up and grab one. So you can grab one of your choice. Orkan, you get the first choice. So you can take this whole batch. Yeah, Annie, come up. Everybody who's doing a breakout session, come on up. So you're going to grab one of those, okay? And then what's going to happen is, for those of you that are interested in joining each of these respective breakout sessions, you will follow the person with that particular pole and that stuffed animal to whatever room you want to go to. Again, there's a lot of space for breakouts. We have space in the auditorium over there. In the lysosome, there's a bunch of space. You can stay in here and have your breakout wherever you want to go, and then have your session. I shared with you all via the Google group a Google shared folder that says session folders, I think it's called, something like that. You guys get this email from me? Yes? Okay. So basically what we're going to do is, inside the session folder, so there's one for today and there's one for the BioArt and Design session, you would create a new Word document, put the names of the people that are there, the time, and before you start your session, assign a note taker and assign a report back person. So this is how we're going to do our documentation for all of these sessions. Somebody's going to take notes, record all the stuff that happened in that conversation, and then on Sunday, we're going to do a report back of all the sessions. Okay? Does that all make sense? Yeah? All right, awesome. So before we break up and do the breakouts, because we're running a little bit late, lunch is going to start around 12. So for the breakouts, y'all can do your breakout until, say, 12:30. Or if you want, if lunch is already there, you could grab your lunch and go have the breakout, however you want to do it. We had the family photo scheduled originally at noon, but because of the scheduling issue, I think we're going to just do that tomorrow. Okay, we're going to do the family photo tomorrow instead. So lunch is going to, or the next session is going to start after lunch at 1 p.m. It's going to be right back here. So Marshall Gans, who was unable to join us yesterday, we're going to try to Zoom him in from DC again today at 1. So actually, Adeline, we're going to do your session after his talk at 1:15. Okay. So a couple quick logistics. The mugs are all ready for you, so when you go out to lunch, you can grab the mugs. They're over by the info point. Again, Staples, we have like we don't like Staples anymore, so don't go to Staples. They're like de-sponsored. I don't even know what that word is, but they kind of messed up the logo and whatever, but still, take your mug. 2, there's now a guest book also at the info point, so please sign our guest book. It'd be really, really great to get some messages from you all. And that's it. That's all I have to say. So without further ado, Orkan's got his design.bio session. We've got speculative design and more, and biodesign challenge. And then Adam, what's the title of your session?
Genetically modified orgiastics.
Genetically modified orgiastics. Okay, so if you're interested in any of these sessions, please find your session leader. They've got, you know, these awesome stuffed animal microbes. I keep saying stuffed animal microbes. That's a weird thing to say. And please join them. So go, have fun. And another round of applause for everybody from BioArt and Design. Thank you all very much. Oh, and anybody that's part of the economy, ecology, and what's the final E? Environment session, Adeline is your session leader. She wants to talk to you really quickly. So anybody that's part of the next session, the 1 o'clock session, please find Adeline. She's right over here. Thank you all.
Hey, 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 hey. Check 2, 2. Hey, 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 hey. Check 1, 2. Hey, 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 hey. Check 2, 2, 2. Hey, 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 hey. Hey. Hey, hey. Hey, hey. Hey, hey. Hey, 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 hey. Check 2. Hey, hey. Hey, hey. Hey, 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 hey. Hey, hey, hey. Check 2, 2, 2. Good morning, good afternoon. Welcome, welcome. All right, live stream's good. I just need to wait for him to connect. Okay, beautiful. Good, how are you?
Good, thanks. I've got a talk later on today, and I haven't given you guys my slides yet.
Do you have it on your USB slide?
I have it on my laptop.
Okay.
I might have a USB stick somewhere.
That would be great. If not, we can. I would prefer to get it on a thumb drive.
There's a hi. How are you? How you doing? Good to see you. You would be the first of the speakers to upload your slides, but there is a Google Drive link
Okay.
that I was just